Hello, my name is Dr. Eric Stricker, and today we will be talking about the application of the scientific method. This is part of the Biology Lab 1152 taught at the University of St. Thomas. We also have an interactive session in this video, so if you want to participate, you need a permanent marker, preferably in black, a hand sanitizer of any size, rubbing alcohol from 50 all the way to 91% isopropyl concentration, a clear plastic sleeve, a ruler, and paper towels. So let's talk about science. Science is an attempt to understand the natural world. It's done by asking questions and uses evidence as well as builds on observations by multiple people. What is special about science is that it's not constant. It's an ongoing process where old ideas are revised to include the most current understanding. If we talk about biology, which is a subspecialty of science, we talk about the things that are living. It comes from the Greek word bios, which means life, as well as logia, which means that it is a branch of study. A wordly translation would be to speak. Now, if we look at the scientific method, it is a special technique by which knowledge is obtained from the natural world by use of empirical data. Empirical data are the data produced by an observation or an experiment. So there are certain steps to the scientific method. First and foremost, you have a question or an observation that drives you to come up with an experiment. But before you start, the scientific methods include research. You want to find out more about this problem, how to answer it, so that you can form a hypothesis. A hypothesis is basically a statement that can be falsified, and it is based on a rationale. Once we have this hypothesis, we can actually plan our experiment and perform it to test it. And lastly, we collect and analyze the results in order to form our conclusion. Most of the time in science, the conclusion leads to even more questions or gives us observations that are interesting, which then puts a full circle to this method and feeds in a constant stream of new innovations and observations and answers to questions. So before we start into our application of the scientific methods, there are specific scientific terms that are not only important to know, but are also handy in order to design correct and the most accurate experiments. So these are the variables that we're going to talk about: dependent variables, independent variables, standardized variables, controls, as well as replication and sample size. And I want you to just pause this video, and if you haven't delved into that, look up what each of this type of variable or word means, and we will continue in a second with the video to go over the content. Okay, now that we're back. Dependent variable. So the dependent variable is in essence what the investigator measures or counts or records. It could be light, it could be plant growth, it could be anything that is actually output for an experiment. Most times it's collected in tables and can be calculated. When we are looking at independent variables, this is what the investigator deliberately varies during the experiment, or in other words, the thing of which the effect is actually measured. If you're interested in a medication, the medication becomes an independent variable as it is altered, maybe in concentration. Maybe one group gets it and one group doesn't. Now, we also need, when we conduct experiments, standardized variables. Standardized variables are factors that are kept equal in all treatments. They are very important so that we can compare different experiments, such as we want to use the same reagents, we want to use the same supplier sometimes for reagents, we want to use the same circumstances, maybe even the same day or the same time of day to measure something, to really keep it as much constant as we can. Now that we have standardized experiments, we also have a subcategory of variables in essence, which are the controls. The controls are the treatment in which the independent variable is either eliminated or is set to a standard value. And lastly, in order to get really accurate data and good predictions from our experiments, we want to replicate our experiments. So replication increases the confidence and reduces the natural variability. A sample size of minimum of 3 is needed in order to perform statistics. This has to do with the fact that only when we have 3 samples, we have a mean and we can calculate what the variation away from the mean is, which is called standard deviation. And it becomes clear the more we test, the more accurate we get. So if we want to flip a coin and I flip the coin only 1 time and I say heads, well, it doesn't really tell me if the coin always lands on heads or not. If I throw it 2 times, I'm getting a better idea, but it could still be happening to heads. And now if I throw it 10 times, I will be more getting an idea if it really is a fair coin that flips 50-50 or if it's unfair. So now that we defined our variables or our terms, let's look at our first experiment involving the scientific method. So I want to start with a story. So this is a real life example. So one day I was babysitting my baby cousin, and I didn't really want to, but I just did it because I loved him. And so mom and dad left, and we had a good time, and I stepped for a moment to the side, got distracted, and when I came back, he drew with permanent marker all over the place. It was on the windows, it was on the leather couch, it was on picture frames, it was everywhere. Well, what to do? TikTok to the rescue. I remembered that there was a TikTok video that talked about how to remove permanent markers. It said to use rubbing alcohol or hand sanitizer if you have it in hand. But knowing that some of the TikToks are just looking for views, I wanted to really experimentally test it before I tried it out on the expensive leather couch, before I messed it up even more. So the first thing that I had to come up with is the question, which is, can you remove permanent marker from surfaces with rubbing alcohol? So now comes your turn, the same thing that I did. So I want you to research to find out what the main ingredients in rubbing alcohol are, or in hand sanitizer for that matter. Find out what permanent markers contain. And then number 3, I want you to see and look up what a solvent is. So I give you a few seconds. You can pause the video right now, and we're going to continue in a second. All right, let's have a look at what we found. So first foremost, rubbing alcohol. So rubbing alcohol, the main ingredient is either isopropyl alcohol or ethanol, which makes the main part of this liquid. Permanent markers, on the other hand, they contain multiple ingredients. So number 1, we have the dye that makes it actually visible. And then we have alcohols such as ethanol, sometimes even isopropanol, depending on the marker. And if you have a look, I don't think mine says it right here, but on the package, it should be written too. Now, when we talk about permanent markers, we also come across the word solvent. A solvent is the thing that keeps the dye liquid. And the special thing about the solvent is, once the dye is transferred onto a surface, the solvent evaporates and leaving behind the dye that's permanently attached to the surface. So in our case, the solvent would be the alcohols, and the solute, if you want to, is the dye. So now that we have that information, we can think about our hypothesis. So what's a hypothesis? A hypothesis is a statement that must be testable as well as falsifiable. Very important in science is that we really have it falsifiable. So it must be possible to contradict the hypothesis. For example, when we say there is a ghost, well, that's not really falsifiable. So therefore, it's not a scientific hypothesis. So for your Now it's your turn to come up with the hypothesis. So I give you a moment. Okay, let's see what we came up with. So first and foremost, we realized that, or had an idea maybe, that by adding the solvent again, it might help removing the marker. So I hypothesis in one sentence would be permanent marker can be removed from surfaces with rubbing alcohol. As simple as that. So now we come to the part of designing the experiment, to planning and performing it. But before we perform it, we really want to think about our different variables that we talked earlier about. So number one, we want to have a controlled experiment. There's different types of experiments that we can do, but for our case, a controlled experiment is the best. That is where a test group and a control group are compared. So in the end, we have to define a control group as well as our test group. And we want to also think about the variables, the independent and the dependent ones. As a repetition, the independent one is what the experimenter manipulates the variables that is different between the control and the test group. The dependent variable is what changes as a result of the independent variable. So that's what we in essence measure, right? So I give you again a few seconds or whatever time you need, so you can pause the video right now, and then we can continue and talk about what I came up with and if that matches what you did write down. Okay, now that you had some time to think about these variables, let's have a look at what I came up with. So for the control group, I would actually suggest some water, because water doesn't have any isopropyl in it, so it would be a good control group. And therefore, the test group becomes the hand sanitizer, the rubbing alcohol, whatever you have at hand. If we look at our independent variables, so the independent variable would be removal of marker. So we can maybe say, yes, it got removed, or no, it didn't get removed. That's what we measure. When we look at our dependent variable, that would be actually the different sanitizers. I told you at the beginning you should get a little bit different sanitizers. Maybe they make a difference. Okay, now it is your turn to collect the data. So what I want you to do is I want you to take this clear foil, make a few lines or marker signs, or maybe even dots if you want to, onto this clear slide, and then take different kinds of rubbing alcohol. So I have 50% here, I have 91% right here, and then I have also just some hand sanitizer, which I don't even remember how much is in there. It's also old. Maybe it's lower. It's at 62%. So let's try that. You can add it to some paper towel and try it out, and then we will be back in a second to look at the results. I want to encourage you to write that down on a table, and let's say we try out every sanitizer 4 times. So I want you to make 4 dots or 4 lines for every sanitizer and write down if it got removed in each of the incidences. Okay, now that we collected our data, we can have a look at what the results could have looked like. So therefore, I actually have linked in the video or sent directly to you an Excel sheet that has trial 1. That is where you can put in the name of the sanitizer and then the different results. And it will generate some really nice graphs. So let's have a look what it can look like. In our past experiments that we did it, we had one group of water and 6 different sanitizers with a few different students. Every student, one group had only 2 people, so they only did it 2 times. Some people had 4 groups, so that's why there's a different number of repeats. So let's have a look. So we had one group that actually with elbow grease got the marker off with water, and then we had different sanitizers. Sanitizer 1 didn't do well. Sanitizer 4, 5, and 6 did pretty well and removed it. So we now have to convert this into numbers, because plus and minuses are good and fair, but really calculations and statistics we can do with numbers. So I'm gonna give it a 1 if it was removed and a 0 if it wasn't removed. So in your table, you will actually see that that automatically transferred when you did plus and minus. So in order to do in the Excel a plus and minus, you have to type in plus and then press the return key. Otherwise, if you click away, it will try to link the cell. So just as FYI. And what you see here on the right, and let's see if I can even show you the mouse here. Yeah. On the right is the sum of the experiments. So here I had 1, and then the trials, that's N, and then in how many percentage of the cases I actually had removal. So that was B25. And I did the same for the sanitizer. So 61% of the sanitizer cases actually removed it. And I also calculated a standard deviation. As you can see here, one time as a decimal, one time as a percentage. It's in essence the same thing, and we can plot it together. So standard deviation tells us, again, how much variation was in there. And if we put this on a graph, it will look something like this. So for example, we have a bar graph. On the x-axis, we have our control and our test group. And then on the y-axis, we have successfully cleaned spots. And you see very quickly that even though the test group was more successful, I wouldn't say that that was very clear, because what we call the error bars, which represent the variation, they show us there is a lot of variation. They are overlapping. The control groups could, in essence, be the same. Just by chance, we got one that only in 25% of the cases removed it. So if we look at this, what would be our conclusion? Can we really conclude that hand sanitizer removes rubbing alcohol, or are we missing something? I mean, first of all, I would definitely say sanitizer does not remove permanent marker stains reliably based on our results. But what are we missing here? Let's go back at the different variables that we talked about. So we designed and performed an experiment, and I talked at the beginning about standardized variables. So every group must be exposed to the same conditions. Standardized variables were the one thing we actually didn't have defined at this point. And as I mentioned, if you have better standardized variables, you also get more confidence that changes are due to the independent variables alone. So standardized variables, I want you to think about something that we can standardize, something that we can keep the same between all the sanitizers. I give you a moment to write that down, and then we're going to talk about what would be some good ideas for standardization. Okay, now that we're back again, let's have a look. So my first and foremost thing was to really, that's why I gave you a ruler, keep the length of the permanent marker line the same. That way we have the same amount of ink, the same amount of dye that we have to actually remove. It would also be very good if we can somehow keep the amount of sanitizer the same. So maybe we can grab a spoon and put the sanitizer on a teaspoon, and we basically have 1 teaspoon of sanitizer that we then transfer to our paper towels and then try to rub away. But we also realized, and I mentioned that, some people actually can use a lot of elbow grease to use water to remove it. So let's standardize the pressure we use. And I would even say minimal pressure, just adding the alcohol and just slide it one time over. And then we can also standardize the number of times back and forth wipes. The longer I wipe and rub at it, obviously, the easier it comes off. So let's say we do, 5 or 10 like back and forth for every section. You can choose that for yourself. Just write it down. And then we can start our experiment with our standardized variables. But there's one more thing that we didn't consider. And that is, I mentioned to you, there's different amounts of isopropyl in these different sanitizers. We already defined our control groups with 0% and our non-control group with more than 0%. But I would, for our next experiment, actually split them apart. So let's do every sanitizer and write down what percentage of isopropanol they have, and maybe we can find a trend or a difference. And if you want to, you can do it 4 times as a repetition for every concentration, or even more or less, but we want to keep it at least 3. So in our case, it would be good for 25%, 50%, 70, 91%, and in our control group 0%. The 25% you can just easily get by taking the 50% and dilute it 1-to-1 with water. So maybe just like a small cup, a little bit of water in it. But now that we have our additional standardized variables, and especially because we now have also our different concentration of isopropanol, we want to come up with a new hypothesis. And I called it higher concentrations of isopropanol can remove permanent marker better. We don't even have to quantify yet if we don't have an idea what percentage is the different making point. But I would definitely say we should define this hypothesis in this way for. So let's collect again our data, and I give you a moment to collect your own, and then we can have a look at the data that I collected here. So let's have a look. So in this time, you see all of a sudden every single water control group was not able to remove it because we standardized. And then when I looked at the sanitizers, I ordered them by isopropanol content, and I saw that the 50%, half of the times I was able to remove it, and then 70% I was able to remove it all, as well as 91%. So now if I plot this again with the standard deviation, the mean for every single, so going back one more time, maybe to keep it simple. So here I have the isopropanol content and the percent, which is the mean of removal. So in 50% of the cases, I was able to remove it for 50% isopropanol, and 0% of the cases for isopropanol content. So if I now plot this with a line plot in Excel, and you find like really great tutorials online if you have struggled with this, you want to plot the isopropanol concentration on the x-axis, so vertically, horizontally, sorry, and vertically the successfully cleared spots again. And what we see very nicely here is that we have this trend line, and we even have a 50% point at 50% where we have removal half the time. So in this case, we can conclude that at least 70% of isopropanol is required to remove permanent marker. Now that we have this conclusion, we have an idea, and we could, for example, now go back and say, well, you said 70%, but we did not measure anything between 50 and 70%. So why don't we just go back and measure 60%? Is that enough? Or 55? I mean, we extrapolated, which is basically meaning we connected the dots, and we would say that in 80% of the cases, it would be removed for 60%, but we don't know for sure. So we could just like come up with new experiments to refine it furthermore. And one thing we also didn't take into consideration is some of these hand sanitizers have other things in them that might help or prevent it from removing. So to standardize even further, we can use really the same brand. We can use the same additional ingredients to do this experiment. Overall, I hope this helped you to understand the scientific method. We went all the way from finding our problem and asking our initial questions to doing research on the problem, finding out about solvents, and get an idea and forming our hypothesis. Then we came up with an experiment that had our independent and dependent variables, as well as defined standardized variables. We defined a control group, which was our water, and our sanitizer as the treatment group. And then lastly, we also had multiple repetitions, which made us more confident. And we had variable numbers of concentrations of isopropanol. Then we tested that whole thing, plotted our data on Excel, and had started to have an idea if this really worked. which gives us the conclusion. Usually you would now go back with the conclusion and look if this makes sense again with what other people observed. I told you at the beginning that other people define science and that it has to be multiple people that observe it. So in order to do this, we either have to find somebody who did the same thing, or we have to write it down so that somebody else can do it. And if they can do it and they get the same results, we can be really confident that we did a good job, had great rigor, and chose our variables in our experimental design good. So with that, we're at the conclusion also of this short video. I hope you had some fun and learned a little bit about it, and I see you next time.